Hello, my name is Michaela Rodriguez, and today I will be presenting my project titled Determining How PSAP Induces Mitochondrial Dysfunction. Again, my name is Michaela Rodriguez, and I am a senior at UT Permian Basin pursuing a Bachelor of Science in Biochemistry. I am currently studying in the Neurodegenerative Research Laboratory of Dr. Shumen Shu, focusing on Alzheimer's disease and ALS using a live mouse model. Dr. Shu's lab focuses on understanding the cellular and molecular biochemical mechanisms involved in neurodegenerative disorders and diseases. Presnicillin-associated protein, or PSAP, has been identified as being a pro-apoptotic mitochondrial protein. PSAP has been observed to induce programmed cell death, or apoptosis, implicating the pathogenesis of multiple disorders like ALS and Alzheimer's. This project aims to determine the way in which the protein PSAP induces mitochondrial dysfunction, leading to programmed cell death or apoptosis. This was accomplished using a cell line, HEC293, which stands for human embryo kidney cells, which have been stably induced by a plasmid that expresses PSAP protein. Upon addition of PON-A to the HEC293 cells, the expression of PSAP was induced, ultimately leading to apoptosis. In addition to the cellular and molecular components of this project, animal behavior and welfare was also assessed throughout the 10-week duration of this project. This component assessed the grip strength of mice, motor ability, body weight, and overall welfare and well-being of the animals on a regular basis. It is with great hope and enthusiasm that my sole mission in pursuing research science is to improve not only the lives of the patients and individual humans impacted by these disorders, but the caretakers, families, and loved ones of these individuals as well. The HEC293 cells were previously stored in liquid nitrogen, and then the culture media was also previously purchased. And I used DMEM, which stands for Dulbecco's Modified Eagle Medium, FBS, which stands for Fetal Bovine Serum, and Streptomycin penicillin. The inducer, ponosterone A, or PON-A, was purchased from Thermo Scientific, but was distributed by Enzo Life Sciences. Additionally, the power supply used for my Western blots were purchased from Biorad and funded for by the gracious ALSAMP SRA program. With this stipend, I was also able to purchase a kaleidoscope molecular marker that I used for my Western blots. The cells were regularly observed under the microscope, and culture media was changed every 48 hours as to ensure adequate health of our cells. Once seeded, the HEC293 cells were subject to the addition of 20 micromolars of the PON-A inducer at a 1 to 1,000 dilution with DMEM. Subsequently, the samples were collected at 0, 2, 4, 6, 8, 10, 12, and 24 hours. This was accomplished using a gentle cell lysis buffer and then stored at -80 degrees Celsius until ready for Western blotting. Once all samples had been harvested and collected, I could begin my Western blotting. Figure one displays a general overview of the Western blotting process. Using the harvested cell lysates, I first sonicated my cell lysis. And then quantified the concentration of my proteins using a BCA assay. Using the BCA results, samples were prepared according to dilutions. Using the BCA results, I calculated appropriate dilutions of proteins. Following the BCA assay, I calculated the appropriate dilutions for my samples and then denatured the protein samples by boiling them in water for 10 minutes. While my samples cooled down, I began the process of setting up my gel electrophoresis apparatus and setup. The first step is to create your polyacrylamide gel pages, and each polyacrylamide gel page, or each each gel membrane. The first step was to create my polyacrylamide gel membranes. Each of these membranes is specific to the protein of interest. The first step of setting up my apparatus was to create my gel membranes. Each gel membrane has a specific percentage of polyacrylamide in it, dependent on the protein of interest and its molecular weight. After the gel electrophoresis, I transferred my proteins onto a PVDF membrane and then blocked them in a milk blocking buffer for 1 hour. Following the blocking period, I began protein detection using a primary and a secondary antibody. Next, I will discuss my experience handling the mice and assessing their overall motor ability and welfare. Typically, I worked with about 8 cages of mice at a time. On the days that I weighed my mice, I also did an animal welfare check, which focused on conditions related to the hydration, appearance, activity, and possible clinical signs of illness in the mice. After taking my grip strength measurements, I allowed the mice an acclimation period of 1 hour. Following the 1 hour acclimation period, I placed the mice on the rotarod apparatus. This was done only once a week, and this was done only once a week with 5 to 7 days of rest in between each trial. The preliminary results of my Western blots reiterate the hypothesis that upon the addition of PON-A, HEC293 cells will induce the overexpression of PSAP protein. This was confirmed using the Western blot methodology to detect proteins PSAP, PARP, and caspase-3, all hallmark to the apoptotic pathways seen in Alzheimer's and ALS. Not enough data was collected to make inferences regarding the behavior of mice. One consideration to take into account is the time that goes behind handling mice, or handling animals in general. In conclusion, neurodegenerative disorders are known to have complex and intricate mechanisms involving a multitude of interrelated cells and molecules. These mechanisms have continued to evolve with time. Therefore, it is imperative to continuously study and understand the mechanisms in which cellular and molecular dysfunction occurs. Lastly, I would like to go into my acknowledgements. This project received funding from the esteemed and prestigious LSAMP Summer Research Academy, serving undergraduates aspiring to attain careers in the research sciences. I would like to give special thanks to Dr. Mika Montes, the LSAMP director at UTPB, Dr. Xu, my research professor and mentor, and the Biomedical Research Center at UT Permian Basin for the continual support and guidance throughout my undergraduate education. Thank you.